Hi everyone and welcome to the summary suite. This video is on gram staining in bacteria. In this video, we'll be discussing the basics of gram staining, why it's important, and how to perform it. So, what is gram staining? Gram staining is a laboratory technique used to classify bacteria into two main groups, gram-positive and gram-negative. It was developed by Danish bacteriologist Hans Christian Gram in 1884 and remains one of the most commonly used staining techniques in microbiology today. The gram stain works by using different types of dyes and alcohol to stain the bacterial cell wall. Gram-positive bacteria have a thick layer of peptidoglycan, which retains the crystal violet stain and appears purple under the microscope. Gram-negative bacteria have a thin layer of peptidoglycan and an outer membrane composed of lipopolysaccharides, which do not retain the crystal violet stain and instead take up the counterstain, safranon, and appear red under the microscope. Gram-positive bacteria are more commonly associated with infections such as streptococcal pharyngitis, also known as strep throat, and staphylococcal infections, while gram-negative bacteria are more commonly associated with urinary tract infections and pneumonia. Now, let's go over the steps to perform a gram stain. 1. Obtain a bacterial sample, either from a culture or from a clinical specimen. 2. Place a small drop of the bacterial sample on a clean slide and spread it evenly. 3. Flood the slide with crystal violet for 1 minute. 4. Rinse the slide with distilled water. 5. Flood the slide with grams iodine for 1 minute. 6. Decolorize the slide with 95% alcohol for 10 to 15 seconds. 7. Counterstain the slide with safranon for 1 minute. 8. Rinse the slide with distilled water and blot dry. And that's it. You can now examine the slide under the microscope and determine the gram status of the bacterial species. In conclusion, gram staining is an important laboratory technique used to classify bacteria into two main groups, gram-positive and gram-negative. It is useful in identifying bacterial species and determining its pathogenicity. With the steps outlined in this video, you should be able to perform a gram stain with ease. Thanks for watching and don't forget to like and subscribe.